Okay, my clock shows top of the hour, so let's go ahead and get started. Welcome, everybody. I'm Denise Ellsworth. I'm with the OSU Department of Entomology and OSU Extension here in Ohio. I'm excited to have everyone here together for our second session of the day on the idea of bumblebees from the western part of North America. Just a few words. I won't take as much time as I usually take at the beginning, but I do want to tell you a little bit about the Zoom platform in case you're not familiar with our Zoom format. So your audio and video are turned off. You don't have to worry about that. We do use the Q&A box, and so that's where your questions will go. Don't rely so much on the upvoting because we couldn't clear out the questions, but just go ahead and put your questions in, and we'll try to filter through those questions to ask Lincoln at the end. Don't put your questions in the chat box. They may get lost. For now, we still have the chat box open for your city and state or city and country. Once I turn the floor over to Lincoln, we'll turn that off so you don't have that chat interruption on your screen. We will be recording this session, and this will be a separate recording from the Eastern session, so you can go back in and catch one of those again on the recording. Just let me double check. Don't use the hand raising. We don't use that. I told you about the live transcript, so I think we're good to go. So again, welcome everybody. It's my pleasure to introduce Lincoln Best, who is the lead taxonomist for the Oregon Bee Project, a big project out of Oregon State. Many of you are involved, I think, with the Atlas or have seen some of the materials and videos produced out of that project, trying to document some of the 650 plus species of bee there in the state. Lincoln also has a strong background in bee biodiversity. He studied across the globe about bees, but does a lot of teaching Mastermilitologists, volunteers, community scientists there in Oregon. And so really wanted to have him come in and give us a good background on identification of those Western bumblebee species. So, I'm sorry. So I wanted to mention the website. So this is where the recordings will be. Some of those videos, I included some materials from that from that Oregon Bee Project. Some of Lincoln's identification videos and other materials there on our website. So again, Lincoln, thank you so much for being with us. I'll pass the podium over to you.
Well, good afternoon now to everyone on both coasts. Thank you everyone for coming. It's amazing to be able to get a few hundred people in the room to do something like this. I'll be talking about identification strategies for Western North American bumblebees for about the next hour. Thank you for that wonderful introduction, Denise. You all can follow me on all these great social media platforms at Bees of Canada. You can hear all my political views. First off, I'd like to thank Ohio State and Denise for arranging this and for hosting me. Thank you to project collaborators, the Oregon Department of Forestry, Oregon Department of Agriculture, who sit on the advisory panel for the Oregon Bee Project, the umbrella project for everything we're doing here. Thank you to all of our Mastermilitologists and also the College of Forestry and Jim Rivers and colleagues who have supported the development of some identification resources through the Fish and Wildlife Habitat in Managed Forests research program. Of course, thank you to the Oregon State Arthropod Collection. And this morning for 3 hours, thank you to Leah Swanson for her technical assistance. Now, I can't move on without saying a big thank you to everyone that's part of the Oregon Bee Atlas, our statewide initiative to document the several hundred species of native bees in the state and their thousands of flowers species that they visit. If you're interested in becoming a Mastermilitologist, you can Google your way over to our website and add your name to our waiting list. I do believe that we have an ongoing intake now of Mastermilitologist apprentices. The program is an online stepwise program, very much like a Master Gardener's program, where you can learn all about the biodiversity, ecology, and so on about our native bee fauna, including lots of content like this very lecture introducing the bumblebees. So over the last few years, the Oregon Bee Atlas participants have documented around 6,500 netted bumblebees from host plants. And so you can see where all of those occur here. That's a lot of data. And then we have the 2021 material to add to that, which will bring us around 10,000 records for 22 bumblebee species. On top of that, I think our Bee Atlas initiative has identified around 20,000 bumblebees through a trapping program, which gives us much higher resolution community data on the bumblebees in the different places in the state. This is what our bumblebee fauna looks like in Oregon right now. We've been able to detect 22 species, and there are a few historical records in red for species we have not been able to redocument. And I've recently just crossed Turgides off the list because I suspect it's likely just old Occidentalis records that were recorded as Turgides. Now, on top of this, I've been looking at bumblebees for around 15 years, and I've had the opportunity to identify a few 100,000 specimens. With other project partners in Oregon, we've probably worked up around 25,000 records. In British Columbia, around 40,000. And then in central Alberta, south central Alberta, around 60,000 through the University of Calgary. So it's a lot of Western bumblebees. I like bumblebees quite a bit. Of course, Oregon just doesn't have a program. There's lots of programs about bees now. And so in Washington State, consider joining the Washington Native Bee Society. In British Columbia, consider becoming a member of our Native Bee Society of British Columbia. And if you're on the east side of the Rockies in Canada, consider joining the Alberta Native Bee Council and working with them to conserve our native bee faunas and to learn more about these amazing critters. Now, this talk will be broken into 3 broad parts. And these parts kind of reflect the 3 stages that you will move through while you learn to identify bumblebees. So as you learn, it's good to start generally with just the color forms and going through the field guide and looking at the geography and habitat. Then you'll move on to learning some microscopic characters, and this will help you place your bumblebees into different subgenera. And we have several subgenera of bumblebees in North America, which is another way to coarsely break them down. And then finally, you'll be able to combine all this information and use the key to species to identify your bumblebees to species. Now, the primary resource that I advise people to use is Williams et al. 2014 Bumblebees of North America. If I know many of you will have a copy of this right beside you right now. If you don't have one, it's available as an ebook. And so it's nice to have a field guide, but it's also really nice to have this book on your phone in your pocket all the time. And so you can go and get this from Kodo right now as an ebook. And that is really nice to have. Of course, we have a picture of Paul Williams here as well. Other great books, for those of you in the East, you can already get Olivia and Joseph's new Common Bees of Eastern North America. And they've put out a poster of North American bumblebees, which is awesome. So go get yourself a copy of that as well. So just to elucidate a little bit more on these 3 stages, when you're using the color forms, the geography, and the habitat, you'll use those 3 pieces of information, and then you'll flip through the different species accounts in the field guide. And while you do that, you'll make a short list of which species your bee could possibly be. And that could be a bee specimen, or it could be a photograph of a bee that you took in the field that day. And by using the color form, the geography, and the habitat, you're able to delimit the total list of bumblebees in North America down to maybe 3 to 5 species that are in the area that your bumblebee came from and that have a similar color form to the one you're looking at. And also, as we saw in the previous talk, bumblebees like Bombus vagans, which may be closely associated with forests, you can use habitat to inform your choice as well. The second stage, we'll be using basic, or I should say fundamental microscopic characters to sort your bumblebees into different subgenera. And then, of course, finally, we'll combine all of this information and use it in the species key to try and get the ultimate identification on your critter. We're not going to do that yet, however. First, we have to learn how to assess the color forms. And even before that, we need to learn how to recognize a bumblebee. So bumblebees are large, they're very hairy, moderately fast flying, and the females of pollen collecting species always have a corbicula, and the corbicula or corbicular scopa is the pollen collecting basket. This is unique to a few different groups of bees, and in North America, that is only the bumblebees and the honeybees. And so if you see a bee with a corbicula, which is a mostly hairless, flattened, widened tibia, where it's packing its pollen, you can be certain that you're looking at a honeybee or a bumblebee. And of course, a honeybee is not nearly as hairy on the abdomen or as large and fat as a bumblebee. And so that's kind of the easy general way to recognize a bumblebee. Specifically, you would look for the lack of a jugal lobe, but we're not going to do that. So here's another potential bumblebee. If we take a look, we can see, sure enough, we have another corbicula. And so that's good enough to convince me, at least, that this is a bee in the genus Bombus. Likewise with this one, it's another bee with a corbicula. So again, this is another female Bombus and a pollen collecting species. Of course, there's always exceptions, and we do have a small number of cuckoo bumblebees which lack a corbicula, and so that hind leg will be mostly hairy. But there are always exceptions when you're dealing with biodiversity. So finally, we have this bee, and if we look at the hind leg, we see that it is very hairy. And so this is a bee that looks like a bumblebee, but does not have a corbicula. So as you proceed through learning the bumblebees, you are really going to want to start with looking at those color forms. And it's not as easy as it might first appear. So let's take a look. You'll have to learn how to transcribe the colors on the actual bumblebee that you see onto one of these color form templates. On the left, we have a template for a female, and females have 12 antennal segments, not pictured in the template, and they have 6 tergites. The male template on the right, you can identify male bumblebees by the fact that they have 1 additional antennal segment compared to the females, as well as 1 additional tergite. And you may be asking yourself, well, what is a tergite? And we'll look at that in 2 slides from now. So generally, insects and bees in particular have a head, a thorax, and an abdomen. And scientifically, we refer to the thorax as the mesosoma and the abdomen as the metasoma. I know sometimes people have trouble remembering which is which. And if you recall that S comes before T in the alphabet, you'll be able to remember that the mesosoma comes before the metasoma on the bee's body. So here we see what the tergites are, and the tergites are the top of the abdominal segments. And so on the left, this female bee has 6 exposed tergites. 1, 2, 3, 4, 5, 6, and the male has 1, 2, 3, 4, 5, 6, 7. Really easy to count those as the bees flying around a flower. But in a picture or in a specimen, you have a much better chance of being able to assess those. And in order to transcribe the color form of a bee onto these templates, you'll have to get a general idea of where those segments start and finish. But fortunately, the colors often are specific to a segment, and so that makes it a little bit easier. Here we have also the front or the face of the bee's head and the top or the vertex of its head, which are important to distinguish. So let's walk through transcribing the color form we see on this bumblebee onto this template. So on the bee's face, we see that the hairs are mixed. On the top of the head, we see that the hairs are mixed, yellow and black hairs. On the anterior color band of the thorax, this one, we see that the hairs are still mostly black and yellow mixed. In this area, we see that they're mostly black in this area. And then on the back of the thorax, or the mesosoma, we see that they're mostly yellow. So that would be this area. On the abdomen, we can count 1, 2, 3, 4, and then the rest is kind of obscured by the angle of the specimen. And so if we look at these different segments, we can see that the first one is yellow. The second two are orange or orangey red. And then this fourth is pretty obscured here. But I would say at least in this corner, there's some yellow or intermixed hairs. So probably both of these corners have yellow or intermixed hairs. So if you flip through your field guide and make a short list, you'll find that one of those bees in the field guide will have a template that looks something like this. And so recall the face and vertex were intermixed hair colors, which in the field guide will be depicted as brownish or grayish. We found that the middle part of the thorax was black and the hind part was bright yellow. And then on the abdomen, at least the first three segments were yellow, reddish orange, and reddish orange. And then the remainder here in our photo was kind of obscured, something like this. And I use these little icons here to indicate the where the photos or diagrams came from. And so almost all of these nice photos here were taken by Thomas Shahan, a world-class macro photographer who was working with the Oregon Department of Agriculture several years ago. And of course, all these color templates are from Paul Williams' field guide. So thank you, Paul, for all these templates. And so we can see here how these approximate one another, but it's not as easy as it might seem. And so this is the black-tailed bumblebee, Bombus melanopygus, melano meaning black and pygus meaning tailed. And so in the field guide, we see these options. And wow, these are not all the same. This one has a black face, as does this one, yellow face, intermixed face. And then we find this bee here, this color morph of the same species, where the red is completely exchanged for black. And so these are all details you get to learn about. So let's do a few more of these. So here we have a bee that has yellow shoulders, black in the middle of its thorax on top, yellow posteriorly on the thorax. Then we have T1, which is yellow. T2, which is here, has a crescent of yellow, and then black around that crescent. T3 is black, and then it looks like, T5 and 4 are quite pale, whitish or yellowish. So let's take a look. You go through your field guide and you might pick up something like this, which is a bee that has that crescent extending onto T2, the second tergite. And so that's fairly diagnostic species. And again, lots of yellow on the thorax, black bar in the middle, yellow posteriorly, and that pale tail. And again, that bee we see in the photo is not identical to this template. And so it takes some practice learning how to transcribe these. And actually in my courses, usually the first step to learning the bumblebees is everyone gets blank templates and a box full of bees, and you actually have to color the templates in according to the bees and then use that to match up with the field guide, which is actually a great exercise to train your eye to look at these different segments. So here's another example. The head is mostly obscured, so we'll ignore it, but the anterior on the thorax looks whitish. The middle is black. And then posteriorly on the thorax here, what we find is there's yellow laterally, but there's this black, almost triangle in the middle that divides the lighter colored hairs, and you can see these black hairs all in here. On the abdomen, we find that T1 is yellow, T2 and T3 are black, and then T4 here is yellow again. So if we flip through the field guide, we should be able to come up with something like this, which is a bee with lots of white on the thorax, black in the middle, and then on that posterior band, we find that it had that light color laterally, interrupted medially by black. And then on the on the abdomen, we had T1 yellow, T2 and T3 black, and then T4 yellow again. And hopefully, if you shortlist some species, you'll find that, well, not too many bumblebees in North America have white on them, so that's pretty helpful. And then only a handful of species have this posterior band interrupted medially, and so that's another good clue. And you will not have extreme success the first time you do this, but if you do this 10 times, 20 times, you'll start to really get the hang of it. And then when you're in the field, you can just crack open your field guide, flip through it, because there's only 46 options for 46 different species in North America, and you'll be able to quickly think, oh, it's got to be either this species or this species. And in a lot of cases, you'll be able to nail it right off the bat. So here's another example. The top of the head is yellow. The anterior band is yellow. The medial band is black. The posterior band is kind of like that last one we saw, where it's interrupted medially with black. And then the abdomen here, it looks like black, 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 and then yellow, yellow, and then this is T6 on the end. So again, you flip through your guide, there's only going to be about 6 options of bees that have a color form that approaches this. And so this is a pretty good representation. What we don't find, however, is a good example where this is black in this area, like I pointed out. And the reason for that is that the color variation among all these bumblebees is on a spectrum. Even within one bumblebee nest, you can have pretty extreme ranges in the expression of their colors. And so it's impossible in a field guide to provide 200 color morph diagrams for one species, and it's actually counterproductive because it makes it potentially more difficult than it needs to be. So another example here, we have a bee. Its head is mostly obscured, and look how long that face is. That's a really long face bumblebee, but the anterior band on the thorax looks whitish. Oh, the middle band is most black, and then we have the posterior band being quite yellow, and then we have T1, T2, T3, and at least T4, and maybe T5 looking quite orange. If you flip through the guide, there's only one bumblebee that has white shoulders and the remainder of the body yellow, and that's the white-shouldered bumblebee, Bombus appositus, which we're actually fortunate here in the Willamette Valley in Western Oregon to have a novel color form for Bombus appositus that lacks the white shoulders. And so it looks even more like all yellow Bombus fervidus with a yellow face. So I think this is our second last example of the color forms. So yellow anteriorly, a black dot, specifically a dot, and then yellow posteriorly. And then we can't see necessarily all of the abdomen, but at least T1 is yellow. Possibly, I can't tell. T1 is yellow at least. So a yellow thorax with a black dot. And if you flip through your guide, you'll find a handful of species where the thorax is all yellow with a black dot in the middle, and Bombus vagans is one of those. Finally, we have this beast photographed by Thomas Shahan. We can see that the face is black, the vertex looks black, the anterior band on the thorax is yellow, the medial band is yellow, but you can see some black hairs in there, and then the posterior band is yellow, T1 is yellow, T2 is yellow, and T3 is yellow, and here T4, T5 are black, and here's T6. So if we flip through the guide, we'll find that this is a pretty good match. Black face, intermixed hairs on the vertex, We have that little bit of black hairs centrally on the thorax. Then we have T1, T2, T2, T3 yellow, and then T4 and T5 black. So the first time you do this, it won't necessarily be easy, but the 50th time you do it, you'll be really good at it. And then you'll become extremely proficient at using your field guide to figure out what these bumblebees are, either from your photographs or even while you're in the field. And if you start just by examining your local or backyard bumblebee fauna, which has a limited number of species, maybe 4 to 8 species commonly in any backyard, you'll be able to start to recognize them. And after 1 or 2 summers, you'll know all those bumblebees in your backyard and the different color forms and the different casts as well. So here I'm going to switch over to the microscope and we're going to do an example. So just stand by for one moment. And so here we have another example of a bee. We can see on the top of the head, the hairs are intermixed, yellow and black. We can see that on the anterior band of the thorax, they're intermixed, yellow and black. We see that medially, the hairs are more black than yellow, but still moderately intermixed. And then posteriorly on the thorax, they're quite yellow with just a couple black hairs in there. On the abdomen, we see that here is T1. It looks all yellow, although, you know, when you're looking at a pin specimen, it's a little crispy than it is clear. So T1 here is all yellow. We see that T2 is at least mostly yellow in the middle, but it looks like there's some black hairs laterally. Then we see T3 is mostly black, but that this orange or yellowish tail is actually extending in the middle. It looks like onto the middle of T3, whereas the lateral parts of T3 are still black. So let's flip back over to our presentation. Let's see if I can do this. There we are. So we should be looking at the color template again. And if we flip through the field guide, you'll see that here is a bee. Oh, here's a bee with intermixed hairs on the face, on the vertex, on the anterior and medial bands. Remember I said that the middle band looked much more black, but still had some intermixed hairs. And then T1 was yellow. It looked like T2 had a crescent of yellow, but maybe some black hairs laterally mixed in. And then, of course, here's that orange-yellow tail that extends medially onto T3, where T3 is black laterally and on the anterior part of T3. And so, again, with some practice, you can become really proficient at being able to take what you see in nature or in the lab and converting it into one of these color template concepts. And that's a really strong method to get IDs on your bees. And of course, this is not just for Western bumblebees, but this is for any bee in North America. Now, once you've kind of mastered being able to apply the color forms and the color templates in the field guide, the next step is really being able to recognize some of those microscopic characters that you can use to really confirm the group identity or the species identity for a bee in a photo or a bee in the lab in a microscopic setting. And so if you learn a handful of microscopic characters, you can confidently place your bee to a different subgenus, and there's about 7 different bumblebee subgenera in North America. And once you do that, then all of a sudden you go from 46 species to knowing that in any particular subgenus, you might have only 1 species or 20 species, and it really is another way to delimit the total fauna down to a subset, and you can combine more information to whittle your way down to a final answer or a final few different answers using the color forms, using the geographic distribution of the species, or even using information of the habitat that the bee was observed in. So in order to identify your bee to subgenus, you can use the first handful of couplets in the key to species in the back of the Bumblebees of North America field guide. What I have done is I took Paul Williams' 2007 papers where he simplifies the subgenera of bumblebees of the world. And I've created a simplified subgeneric classification where I took out all of the stuff irrelevant to North America, and I just narrowed it down to be North America specific. And so this is essentially the same characters that you'll see in the field guide. And these keys to the subgenera are for females and males, and both of these two different keys just have 7 couplets. And so we'll work our way through them. I'll show you some examples of the different characters that people find difficult, that once you learn them, it will help you become an authority at identifying your bumblebees. So here we go. So the first couplet simply asks, does your female bee have a corbicula because it collects pollen, or does it not have a corbicula? In which case, you know it's a subgenus Cytherus, the cuckoo bumblebees. And here we have 4 species of cuckoo bumblebees, which I've included here. So if you recognize your bumblebee as a female, that it's lacking a corbicula, you've already narrowed that species down to these 4 options. And if you include information on their range and on their color forms, you may even be able to narrow that down further. If your bumblebee does have a corbicula, you're going to look at the hind basitarsus, which is the segment on the leg after the corbicula. And you're going to look at this little process that points backwards. You're going to look at it, whether it's short and fat or long and narrow. If it's long and narrow, you're going to continue through the key. And if it's short and fat, in this case, it's going to be the subgenus Bombeus. And in the West, we really just have Bombus nevadensis. We don't really have Bombus oricomus in the far West. And so 2 characters will help you identify this subgenus and this species specific to the West, just by looking at whether your female bumblebee has a corbicula and whether it has this short fat process at the base of its hind basitarsus, pointing posteriorly. So these are all tongue twisters, but we try and keep it as simple as possible. So moving on, if it has the long process and a corbicula, we're going to compare the middle basitarsus, and this is where the basitarsi would go. These are going to be the bee's feet right here, and these are the tarsal claws, just to give you a reference. The head is going to be on this side, and the abdomen will be on that side. So we're going to look at the apical posterior corner, and on some subgenera, it's very strongly pointed. It's an acute angle. And in other subgenera, it's going to be kind of rounded, but not, I would say more importantly, not distinctly pointed. One challenge to this is that this area is often covered with hairs or bristles or setae, and so it can kind of be obscured. Now that said, I'm going to pull one up on the microscope. So I trust I'm projecting the microscope image, and here you can see on this bee, this acute angle, this acute angle right here. And so you want to be able to look right through the hairs, and an easy way to accomplish that is to have a white background that allows you to actually silhouette this structure and see right through those hairs. And so here you can see that this very much looks like what is pictured in the field guide. 45-degree angle, and all of these hairs and setae and bristles need to be completely ignored, which is a bit challenging because you see this one here and you say, oh, wow, it's super pointy. But that's not what we're looking at. We're not looking at this. We're looking at this angle, which is that dark silhouette and almost in the background that you can see. Now let's contrast that to the other character state where we have the mid-basitarsus distal posterior corner, not acutely pointed. So one of the big tricks for this is not always even just learning the characters, but it's learning how to use a microscope effectively to be able to find and get a good look at the area that we're interested in. And so here we go. Use our cheat sheet. And there we go. So this you can see is almost a right angle. And in contrast to the 45-degree angle and the distinct point, I hope you're able to at least kind of nod and agree that, oh yeah, I can see it. So let's head back to the key, and we'll go two different directions, of course. One for the pointy foots and one for the rounded foot. So if you have the pointy mid-basitarsus, you're going to be a Thoracobombus or a Subterraneobombus. And these can be further differentiated by looking at the clypeus and whether there are lots of punctures in the middle of the clypeus. I should say lots of punctures here. You can see all these little divots, or none or few in the case of Subterraneobombus. And this will help you sort out some of those species such as Bombus fervidus and Californicus, Pennsylvanicus, compared to the white-shouldered bumblebee and the boreal bumblebee, Bombus appositus and Bombus borealis. Now, if your bee had that rounded foot, you're going to go on to couplet 5. And keep in mind, we only have 7 couplets here. And you're going to end up looking at probably the tooth. And so here we see that the tooth has a distinct notch. And here we find that the tooth does not have such a distinct notch. And that is that we're looking at the left side mandible here. This left side of the face. This is the labrum. The clypeus would be here. And so looking for a notch or not a notch. Alternatively, you can look at the convexity of the clypeus, but I find that's more difficult than just searching around for a notch on the bottom of the tooth. So of course, if your bumblebee does have a notch in its tooth, it's going to be a Bombus sensostricto, which in the West is usually going to be Bombus occidentalis, but possibly Turricola and hopefully a Franklin's. If it has a long face, it's going to be an Alpinobombus, and you'll find those at very high elevations. So this is one of the characters that comes up all the time, and this is the malar length, the cheek length of the bumblebee, and trying to assess how long is it. In the species key, there's a couple specific couplets that ask, is it about as long as wide, or is it slightly but distinctly longer than wide? And so I'll do my best here to show you how to assess the malar area. So here we're looking at the left side of the bumblebee's face. This is the left eye. This is the mandible. And so here we have the condyles and the articulation of the base of the mandible along the base of the face or cheek. This is the labrum. This is the clypeus. And so what you're going to do is you're going to measure the width of the malar area across those condyles. The mandible articulates. And you're going to measure the length of the malar the malar area or ocular malar area from the middle of the bottom of the eye to the edge where the cheek ends. And so this takes a lot of practice. The first 10 bees you look at, you're going to say, well, that one's kind of long and that one's kind of short, but sometimes that's not good enough to be able to use that character. And in this case, I like to try and imagine a rectangle. Yeah, not the best rectangle, but this is the length and this is the width. So if you imagine that this area is rectangular, in this instance, I would say that this malar area is slightly but distinctly longer than wide. And so if we were down to 2 down to one specimen, and it's either a Bombus sensostricto or an Alpinobombus, like in this couplet, this would definitely be the Alpinobombus. And the Bombus sensostricto would have a mailer length about this wide, much wider than long. So finally, we have two remaining subgenera, and these can be distinguished quite easily by looking at the size and distance from the lateral, here we are, the lateral ocellus to the inner eye margin. If the ocelli are rather small and distantly spaced, this is going to be a Pyrobombus. Pyrobombus has by far more species in its subgenus in North America than any of our other subgenera. And if it has the ocelli relatively larger and closer to the lateral eye margin, it's going to be a Culumanobombus. And Culumanobombus are these species where the cheek is very short. The queens are fairly large, especially Morrisoni, which cruises around the high desert. And I really like this character because it makes it quite easy to differentiate a Pyrobombus from some of these other species in a pinch. So sometimes we get Bombus graziocalis and Bombus impatiens, which can look quite similar. And if you just look at the ocelli, you can tell what it is instantly. And the same goes for Bombus rufocinctus, which has a color form that mimics basically every other bumblebee in North America, I would say. If you just look at the ocelli, you can tell quite quickly that this is more than likely a Culumanobombus. So let's just look at those ocelli closer up. So this is the character for Pyrobombus. This is the lateral ocellus. This is the distance to the eye margin. And we see here that there are about 2 ocellar diameters, give or take, to the lateral or inner eye margin. And then in this other case, we see here is the lateral ocellus. This is the distance to the inner eye margin. And if we were to draw some comparative ocelli in there, we're going to get about 1.5 ocellar diameters separating it from the eye. And on a bee, this looks like the ocelli are very small, and these ones are much larger. And I really like this character. So that's it for the females. If you can learn what kind of Corbicula the bee has, some of these pointy knee characters, whether the tooth has a notch, how long the mailer length is, and how wide or distantly spaced the ocelli are from the eye, you can identify all your bees to subgenus. And that really helps narrow down the species identity. Now let's take a look at this male key. So this is the same thing. Paul put out 2 simplified keys, one for females, one for males. And so I took his key, I chopped it down to North American specific subgenera, and it too contains 7 couplets. And for the males, to use this key, you only look at the male genital capsule, which usually needs to be dissected and is affectionately known as the venus. So in this key, we're primarily going to be looking at the gonostylus, the volsella, and the penis valves, which are paired in the middle. And one way to think of this structure is that there's 4 prongs. There's 4 prongs. The penis valves are on the inside, and the gonostylia are on the outside. So I always like to keep things as simple as possible. And if you have a Cytherus, it's going to have the gonostylus. It's very simple with long hairs. And so as we know, system resources. So as we know, there's 4 species of Cytherus in the West, and this will quickly tell you that it is at least one of those 4. If it doesn't have such a simplified gonostylus, you're going to move on. Here, you can ignore the genitalia characters and just look at the antennae, whether they're medium to long or if they're very short. If the antennae on a male are very short, it's a bombeus. And as we know, in the West, we have Bombus nevadensis, a giant bumblebee. And so you get this to genus and species at the same time because we only have one member with the relevant geography. So of course, if your male bumblebee has medium to long antennae, you're going to look at the penis valves. And here, the penis valves are either narrow in width. So this width here, they're either narrow or they're very, very widened. If they're very, very widened, this will be a Bombus sensostricto. And so you have yourself a Bombus turricula, a Bombus occidentalis, or hopefully a Bombus franklini. With narrow penis valves, and that's these here, just as a reminder, paired penis valves. Often you're looking at the structure right on the end. If your bee has narrow penis valves, then they may be straight, turned out, or they may be hooked inwards. And so some subgenera like Pyrobombus invariably have the penis valves hooked inwards, and it's very diagnostic. So if they're straight or hooked outwards, you're going to have either a Thoracobombus or an Alpinobombus. If you have an Alpinobombus, the bee better be from high alpine or very high latitudes. And of course, here you can look at that gonostylus to see if there is a process, a little spine or not. And that makes it pretty distinct which one you have. Now, if your penis valves turn in and they're hooked, you'll take a look at the gonostylus. And if it's simple, as in this case, or if it's a more complex shape, you'll have a different subgenus. But if it's simple, you're going to have, if you have hooked penis valves in a simple gonostylus, you're going to have a Pyrobombus. My pointer go. There we go. So you're going to have a Pyrobombus, which we know has most of the species in North America. And if you have a turned inward or hooked penis valves and a more complex shape to the gonostylus, you're going to go to the final couplet, which separates Subterraneobombus and Culumanobombus. And once again, looking at the penis valves, having trouble following my cursor. If you look again at the penis valves, you'll find that in Subterraneobombus that they have an angle or a process along the mid-length. And then in Culumanobombus, they're lacking any angle or process at the mid-length. And that's it. It's very, very easy to assess the penis valves, especially on these critters. Now, of course, we've looked at a few different genitalia characters on the males, and we've looked at half a dozen different characters on the females, microscopic characters, including notches on the teeth, the malar length, the size and distance from the eye of the ocelli, and so on. So we've looked at how to transcribe the different colors and different color forms onto these templates. And so this is where you'll integrate all of your skill interpreting all of this information. And of course, to get a final answer for males and use the species key to males, you will need to dissect the male genitalia. And so I'll give you a bit of a genitalia demonstration where we're going to examine the penis valves of Bombus caliginosus and Bombus vosnesenski. Now, these two species are rather notorious on the West Coast. Their ranges completely overlap as far as I can tell, and they look almost identical. However, if you just take a look at their penis valves, they can be easily distinguished from one another, where Bombus caliginosus has much wider valves and Bombus vosnesenski has quite more narrow and pointed valves. And so bear with me for a moment while I switch over to the microscope and zoom in on these valves for you. What you've all been waiting for is This is the genital capsule. Here are the gonostylis, the volcella on the end, and here are the valves in the middle. It is the reproduction is a little blurry, but you can see that they're hooked inwards on the end, and on the other one, hooked inward on the end. And it looks like this hooked part is of a fairly even thickness all the way around this bend and then down onto the straight part of the valve. And so this is, these are what the penis valves for Bombus caliginosus look like. And I'll provide you with a comparison to Bombus vosnesenskii, which has the valves much more narrowed. Stand by. All right. I think that's about as good as we're going to get. So here you can see that the width of these valves, it looks like they're about half as wide as Bombus caliginosus, and these are and much more pointed. So these are the valves for Bombus vosnesenskii. Often you don't have to fully dissect the genital capsule out of the bee. You can actually just tease apart the apical segments in order to just get a view of the penis valves, and then you can move on confident with your determination. Let's just get back to here. All right. Why don't we just move into some questions? I mean, we're almost at my 60, and I might just try and turn it off and turn it on again. And
because there were questions about just kind of fundamentally males versus females. So that maybe you could show the Corbicula and kind of how you, how you can even get that distinction.
That's a good question. So,
and then maybe also talk about the queen. There was a question too, like what is her, the makeup of her leg, and how could you distinguish that?
Yeah, the queen and the worker has the same pollen basket structure, the corbicular scopa, and males have very often much longer and very skinny legs. Are we good?
Yes.
Let me change. Oh, there we go. We're alive. So I just have a few slides left. So as you can see, learning the bumblebees proceeds from fairly straightforward learning how to transcribe and understand the different color forms in collaboration with the field guide, to learning microscopic characters that'll help you place your bee specimen or sometimes your photos into subgenera. Even just the males having very long antenna versus very short antenna can help you figure out what species or what subgenus it's in. And start with baby steps, and just through repetition, you'll be able to learn all the species in your area. And so this little template here I put together a few years ago. These were all the bumblebee species from one particular study in one valley in the Pend d'Oreille in British Columbia. And so if you walk around with this card in the local fauna where these specimens are the only ones you're likely to encounter, it's not too difficult to say, oh, it's one of those, or it's one of the ones with orange on the abdomen, or it's one of the ones with white on the thorax. And over time and repetition, you'll be able to become more proficient. Now, that doesn't make it easy. And so in the field guide, Paul introduces, I think, 4 different mimicry complexes. I kind of combine his complex 3 and 4 into one, and that's all of these different kinds of critters. And so as you become more proficient with identifying bumblebees, especially ones like this in the West, what kind of strategy are you going to take to, if you flip through the field guide, it's like, well, geez, any one of these could pass for any other. And so maybe that transcribing what you see onto the templates isn't going to work particularly well when these things look nearly identical. They're just very subtle differences. And so the way to do this if you're fortunate enough to have specimens on hand, is you can identify them to subgenus. And then again, that helps you delimit the potential number of species you'll be working with. And so here, if we identify them to subgenus, we see all of a sudden, oh, I bet I could guess what some of these are. Oh, I bet this is a Californicus. And I bet this is maybe a weird Rufosinctus. And I know these are cuckoo bumblebees. So in my area, there's only really two that I come across, and one has a more black face. And of course, if you can tell that it's a Bombus sensostricto, you're like, oh, well, this is probably occidentalis, and this one is maybe Bombus franklini. And so by taking those steps, you can ultimately not become just proficient, but you can become an expert by being able to combine all of these different pieces of information, the color form, the geography, the habitat, and these microscopic characteristics to be certain of your bumblebee's identity. So with that, I want to thank you all for your time and of course for your patience, and I'll be happy to take any questions.
Okay, great. Thanks, Lincoln. So so there were a couple questions on that kind of male, female. How do I get them in the right place? What does that Corbicula look like? And I don't know if you have a couple examples you want to just pop under the scope for us. It'd be nice.
I can try. It's
okay. Don't want to crash it, but if
well, no, it should be fine. But the challenge is that if it's my hand holding it, it's going to be waving around a bit. But we'll try. So.
And the associated question was then how a cuckoo would look different.
Are we live on the scope here?
I do see the,
okay.
Yes. The Venus.
The Venus. Okay. So I think we're on freeze frame. No, I'm going to have to get out of that. Unfortunately, I think all my memories get eaten up by having too many big programs running. So we're just going to go back here and I'll just describe.
Sure.
So if you want to differentiate male and female bumblebees, as we know, almost all bumblebees have a pollen basket, just like a honeybee. So the hind tibia is widened and it's flattened and it's hairless in the middle, often appearing polished, and it has a long fringe of hairs on the outside of it that provides some structure to hold the pollen in there. That's, of course, not true for cuckoo bumblebees, but that's a whole other challenge. So male bumblebees, the hind legs will be just very long, like gangly looking, and they'll be very thin. And in addition, bumblebee that has noticeably long antennae is almost always going to be a male, and a bumblebee with short angled antennae is likely to be a female. You can always count the abdominal segments like we saw earlier in the talk.
Okay. Laura wants to know if you know of a publication that combines photos and the color template together.
That's a good question. Combining color photos and the templates. No, I tried to do that in this talk and when I'm teaching, but I don't know any actual publications that do that.
Okay. Robin wonders if you can share the blank template with me, and then I can post it to our website. A little coloring in the works, I think.
Oh, sure. If you email me, I'd be happy to.
Great.
It's a super fun coloring exercise. In our lab course, we'll give people actual bee specimens to translate and color in. But of course, you can do this with anyone, just giving them a photo. And so for teaching younger people, I actually have laminated photos of bumblebees where they can do their best job to translate it onto the template. It's fun. It's good for your blood pressure.
Carolyn just has a comment that the red pen was really great.
Oh, good. I'm glad you think so.
Yeah.
It's super fun for me. T1, T2, T3. Yeah.
Let's see. And so David wonders if you can suggest good microscopes for this kind of analysis, balancing cost versus ease of use. And
absolutely.
Also taking pictures of what you're seeing.
So I probably identified 30,000 bumblebees with an AM scope in the last 3 years. And an AM scope, you can get a trinocular AM scope with somewhere around 5 to 45 times total magnification. You can get one of those for sure for $350 US. The camera will be extra. However, I can't advise you on running a camera on your amp scope because I haven't done it. This setup here is a Leica with a Flexicam C3 on it. So some people do it successfully. I haven't tried it. I just use mine for identifying the bumblebees. And I found that although, they're not the sharpest optics available, the difference in price between a good amp scope and the sharpest optics available is prohibitive. So for $350 and maybe $30 in lighting, anyone can have a brand new scope at their desk. And that it's really the difference between really getting to understand the morphology of bees, which helps you understand their function, because a bee's body is like a Swiss Army knife. Everything has multiple functions. Each leg is different and provides different functionality. And that functionality, of course, is specific to what they need to do, the behaviors, whether it's digging or grooming or combing pollen out of their hair or cleaning their antennae and all these different functions. And so if you have a microscope, you can learn so much more about the bees than you might be able to otherwise.
Okay, so Rachel has a question about workers, noting that they can vary in size a lot within species. She says it's sometimes hard to tell whether she's looking at a queen or a big worker. How do you reliably differentiate those?
Well, there's a few ways. I just kind of guess. So I look at, like I said, tens of thousands of bees. And if you're looking at a large sample from one location, the biggest ones for any species are the queens. But technically, you can dissect the queens to see if they have mature ovaries, because the workers are non-reproductive and they won't have mature ovaries. And so to be very specific about it, you can look at their internal sex parts. If you want to be, a little less obsessive about being correct, you can actually measure the intertegular distance, which is the width between the wing bases. And if you plot lots of individuals of the same species by their size, you will see that the different castes have almost not overlapping sizes. So the queens will have a normal distribution within a certain size range, and the worker caste will have a distribution that is, somewhat smaller and almost completely separate from the normal distribution of the queen size. And so, but that's a lot of work as well. Now you have like micro calipers out and you're measuring all your bumblebees, which I encourage you to do it, but it's not for everyone. And so alternatively, if you have a decent sample of one species, the very biggest ones that are all similar in size would be your reproductives, and everything else below that would be non-reproductive. That said, some bumblebees have workers with like really incredible variation in size. And until you become more familiar with the specific species of bumblebees, it's hard to gauge in the field whether it's a queen or a worker, unless it's a giant, thumb-sized monster bumblebee, in which case it's certainly a queen. But in the West, we have some bumblebee species where the worker size is gigantic. So things like Nevadensis and Appositus and others, their worker size is often larger than the queens of many of our Pyrobombus species. So queen Bombus mixtus is probably smaller than a worker Bombus appositus. And so a lot goes into it, but you can do it.
Kayleen, can you enter in the chat box the scope name? There were a couple questions about that, and I think it would just be easier if you could just put that in the chat box, what you recommend.
So I would love to. If there's anyone here from the Oregon Bee Atlas, if you can pull up our guidance document for the microscope and put the model and link into the chat for me, that would be great. The challenge is that on the AmScope website, they have like 500 different potential configurations. So I can say, you know, stereo microscope with this range of zoom, and a trinocular for mounting a camera. But when you go to the website, that's not going to help you that much. And so, you know, any stereo dissecting microscope with a magnification range of 5 to 50-ish total magnification will allow you to look at bees really well, including most bee genitalia. And then, of course, a trinocular mount is handy for picture taking or streaming online with your friends.
Okay, great. So I'll follow up with you. You can get me
Sure.
that link.
We have a document for that.
The Bumble template, and folks, I'll put that then on our webpage.
And all that kind of information is also available through our Mastermilitologist course. So what kind of microscope you might want to get, how to take care of your collections, all this kind of stuff. Tons of resources, endless amounts of resources are available.
And you said that course is completely online, is that right? Or...
That's right. So our Mastermilitologist course is a Master Gardener-like course that is self-paced, and it is stepwise. And so you start out as an apprentice, and you can ascend to a journeyman and a master ultimately. And it's all online using the Canvas platform, which is a common platform for universities for online learning. And we have dozens of videos like this one about all the different bees, covering, you know, dozens of genera. We have specialist talks on different other bee subgenera. Like, really, if you want to become a hyper bee nerd, that's the place to do it. And it's also the training for our Oregon Bee Atlas. And so the Oregon Bee Atlas has been fairly successful, that we're now kind of exporting the infrastructure and the concept for the project to Washington State, which is starting their own Bee Atlas through the Washington Department of Agriculture and Fish and Wildlife in collaboration with the Washington Native Bee Society. So good job to all of you folks. Idaho is trying to start their own Bee Atlas. And so we're providing that basic bee education, bee sampling education, working with specimen-based collections and how to collect data and everything else. And it's been so much fun. I just love it.
That's really incredible. So I'll post that link as well to that online course. Super. Let's see. I had one other question I think we didn't get to, which was about tongue length, how you can distinguish tongue length in the field.
Just got to pull on it, I guess. So, you know, among all of the different kinds of bees in the world, they're kind of broadly broken down into two groups, the long-tongued bees and the short-tongued bees. All bumblebees are within the long-tongued bee clade. But that said, among our bumblebees, different subgenera can either be longer-tongued or shorter-tongued, and also longer-faced or shorter-faced. And so if you're a bumblebee with a long face and a long tongue, you're probably really good at visiting flowers with long corollas, maybe penstemons or giant mimulus or all sorts of things like that. And if you're a bumblebee, say like the Western bumblebee that has both a short face and a relatively shorter tongue, you're probably, like in my title slide, going to be visiting something like an aster, you know, rabbit brush or thistles or solidago, goldenrod, and that kind of thing, where the resources are much more easily accessible. And so it's a long answer, as short as possible.
Great. Well, thanks, Link. There have been lots of thank yous in the chat box. Folks, thanks if you've been here all afternoon. Thanks for your stamina, your interest in learning more about bumbles, how to identify them. We'll post a recording link as soon as we can. And again, big thumbs up and thank you to Lincoln for your time and all your great techniques of teaching this online. Really appreciate that.
Love it. That was a lot of fun. Thanks for having me.
Great. Thank you. Thanks, everybody.